Hi friends, a hearty welcome to the third session of the chapter Human Reproduction. And today's class we are going to learn the topic Gametogenesis. Among that, first let us discuss Spermatogenesis. You know that what are the events that taking place in the human reproduction? Of course, there are gametogenesis, insemination, fertilization, implantation, gestation, and parturition. So these are the events that takes place in the human reproduction. First, let us discuss gametogenesis. Gametogenesis means it is the process of formation of gametes. Of course, we know that the sexual reproduction requires two gametes and their fusion. Here, the male and female gamete fuse together during fertilization. For that, the gametes should be formed first. So that process is gametogenesis. These gametes are haploid with half of the chromosome number. These haploid gametes fuse to form diploid zygote. These haploid gametes are also produced through gametogenesis, both male and female gametes. Now let's see that the process of formation of male gamete is known as the spermatogenesis, whereas the process of formation of female gamete is known as oogenesis. The spermatozoa and ovum are formed from the primordial germ cells, and the primary reproductive organs, that is the testis and ovary in case of male and female, that are originated from the endoderm. Gonads are endoderm in origin. Now let's see in little more detail about the spermatogenesis. Spermatozoa are formed in the seminiferous tubule of testis, and the process of formation of spermatozoa is known as spermatogenesis. In males, this process starts at puberty. Spermatogenesis includes two stages, that is the formation of spermatids and the metamorphosis of spermatids. The spermatids are formed by three phases or three stages, that are the phase of multiplication by mitosis, second, the growth of these cells, and last, the maturation by meiosis and further development. Now let us see little more in detail what are these three processes leading to the formation of spermatids. First one is the multiplication phase. The primordial germ cells multiplies by continuous mitotic divisions to form spermatogonia, and it forms spermatogonia which are diploid in nature. And this spermatogonium enlarges in its size to form primary spermatocytes. That comes under the growth phase. The spermatogonial cells that undergo first meiotic division are called primary spermatocytes. Primary spermatocytes are also diploid in nature. Now, the primary spermatocytes after meiosis 1 divides into two, and it forms two haploid secondary spermatocytes with haploid number of chromosomes. Then the secondary spermatocytes undergo second meiotic division to form four haploid spermatids. So all these events that takes place in the growth phase. And third one is the maturation phase. Maturation phase is actually the transformation of spermatids to sperm or spermatozoa. Okay, the process of transformation of spermatids to spermatozoa is known as spermiogenesis. During this spermiogenesis, spermatids embed their head into the Sertoli cells. I hope that you are remembering where is Sertoli cells. It is in the seminiferous tubule, inside the seminiferous tubules. There are two types of cells we have learned, Sertoli cells and germ cells that present in the seminiferous tubule. Into that Sertoli cells, the head of these spermatids embedded into it, and it absorbs nutrients and transforms into spermatozoa or sperm. Now let us see the schematic representation of spermatogenesis. It's starting from here, the primordial cells that undergo mitosis, large number of mitotic divisions, and it is multiplying, and spermatogonium is formed. Spermatogonia is plural, spermatogonium is the singular. Okay, spermatogonium is formed, which is diploid in nature. These spermatogonial cells again undergo mitosis, growth, and it become primary spermatocytes. We have already discussed primary spermatocytes are the spermatogonial cells which are going to take part in first meiotic division. The primary spermatocytes undergo first meiotic division and two haploid cells are formed, which are known as the secondary spermatocyte. The secondary spermatocytes undergo second meiotic division. This cell is also dividing into two, and this one also dividing into two. The second meiotic division results in the formation of four haploid spermatids. These spermatids are non-motile. It is like the precursor form of spermatozoa or sperm. It needs lot of transformations. Lot of changes should take place to become a functional sperm. And the process of transformation of spermatids to spermatozoa by differentiation, and this process is known as spermiogenesis. Okay, and let us see the spermatogonial cells, the germ cells, primordial germ cells undergo multiplication by mitosis and the formation of spermatogonium, and again the spermatogonium divides mitotically and form these spermatocytes. This much include in the phase of multiplication phase. Then the primary oocyte, that is the cell which is going to enter into the first meiotic division, it divides and secondary spermatocyte is forming. That also will grow and enlarges in its size, and from the division up to the second is known as the growth phase. Now the maturation phase from the secondary spermatocytes, its formation by meiotic division to meiosis II and forming the spermatids, and transformation of the spermatids to sperm by a process called spermiogenesis. The entire process is known as the spermatogenesis, whereas the transformation of spermatids to spermatozoa is known as spermiogenesis. Now let us see what are the events occur during spermiogenesis. As I told that these spermatids are non-motile, they cannot move because they are not developed for locomotion. It has a nucleus and cell organelles such as Golgi, centrioles, mitochondria, etc. During this transformation, the weight of the gamete reduces. In the locomotory structures that develops, the nucleus becomes compact and the Golgi complex of spermatids give rise to acrosome, and this acrosomes produces some lytic enzymes. Two centrioles of spermatids become arranged one after the other behind the nucleus, that are the distal centriole and proximal centriole. The mature sperms are released from the seminiferous tubule. That process is called spermiation. The process of release of mature sperms from the seminiferous tubule is called spermiation. Now let's see some of the terms that have giving some confusion in you, that are gametogenesis, spermatogenesis, spermiogenesis and spermiation. Now let us see the diagrammatic sectional view of seminiferous tubule. That means a portion of the seminiferous tubule is enlarged here. The seminiferous tubule consists of two types of cells, that are the Sertoli cells and the germ cells. All these are the germ cells, Sertoli cells and germ cells. You can see this is the Sertoli cells that are located this side. This is also, here is the Sertoli cells, another Sertoli cell, Sertoli cells, and all these are germ cells, which include the basal portion, periphery. We can see the spermatogonium, and further development is taking place. Then the primary spermatocyte, secondary spermatocyte, then spermatids, spermatozoa, all these are formed. Along with the Sertoli cells, it is present in the seminiferous tubule. Moving on to the next is the structure of human sperm or spermatozoa. The sperms have mainly three parts: head, middle piece and tail, but in some text the neck is also considered as a part, so we can say that head, neck, middle piece and tail. Now let's see the structure of sperm in little detail. This is the head region of the sperm, and it has an elongated cell with elongated nucleus containing the chromosomal material, and at the tip of this head a cup-like structure we can see, that is known as the acrosome. We have learned that the acrosome, which consists of some enzymes that are necessary for the dissolving or penetrating the egg membrane during fertilization. The entire sperm, that is from the head, neck, middle piece and tail, everywhere it is covered with the plasma membrane. Head, it consists of nucleus, then cytoplasm, and a cup-like structure called the acrosome. On to the next is the neck region. Neck that act as the connecting link here between the head and the middle piece. This region is the neck. It consists of two centrioles. One is closer to the head region, that is the proximal centriole, and other one little away from the proximal centriole, that means towards the middle piece, is the distal centriole. The next is the middle piece. Middle piece is the region that is seen between the neck and the tail. And the axial filament can be seen through the center and both the sides of axial filament. Mitochondria are present. Mitochondria, we know that they are the sources of energy for the locomotion or the movement of this sperm. Last portion is the tail, the axial filament covered by the plasma membrane. So this is the tail. So a typical mammalian sperm consists of head, neck, middle piece and tail. This diagram is also very important. So you please draw and practice this diagram. What I told, that explanation can be given here like a summary. The structure of human sperm, the plasma membrane envelops the whole body of the sperm. It is divided into head, neck, middle piece and tail. The major part of the sperm is the head and it contains an elongated haploid nucleus. As told, the anterior portion has a cap-like structure called acrosome, which is filled with enzymes that helps in fertilization. Neck is the portion that connects the head and the middle piece. It has two centrioles. The proximal centriole found near to the base of the head. It is necessary for the first cleavage, whereas the distal centriole give rise to the axial filament of the sperm. And the middle piece is a portion that connects between the neck and the tail, and its characteristic is a large number of mitochondria are present in it to provide energy for the locomotion of the sperm. And the last portion is the tail, and it is actually an elongated flagellum, the axial filament surrounded by plasma membrane. This structure helps in the locomotion of the sperm. Coming to the next is the hormonal control of spermatogenesis. That means how the process of spermatogenesis is controlled. Spermatogenesis is under the control of endocrine hormones. The hypothalamus that produces a hormone, that is gonadotropin releasing hormone GnRH. And the gonadotropin releasing hormones, it act on the anterior pituitary and it produces ICSH, interstitial cell stimulating hormone, equivalent to LH or luteinizing hormone, and FSH, the follicle stimulating hormone. ICSH or LH stimulate the Leydig cells. You know that what is the position of Leydig cells? Leydig cells are present in between the seminiferous tubules, that is in the interstitial space. Okay. These Leydig cells produce testosterone. Testosterone is essential for spermatogenesis and spermiogenesis. Then the FSH on the other hand stimulates the Sertoli cells, and the Sertoli cells stimulate the process of spermatogenesis. It also develops ABP, that is the androgen binding protein, that helps in binding the testosterone, thereby stimulating the spermatogenesis process. And the Sertoli cells also produce another hormone called inhibin, which checks the excess secretion of ICSH or LH. If that is secreted in excessive, inhibin is released, and this inhibin gives like a feedback, and it suppresses the synthesis and secretion of GnRH and the FSH or LH. Okay, you can see the flow chart and try to understand that. The gonads have the regulation or the control by the endocrine secretions, endocrine glands. Hypothalamus, it produces hormone called gonadotropin releasing hormone or GnRH, and the GnRH will stimulate the anterior pituitary adenohypophysis, and it secretes two types of hormones, that is LH or ICSH. The LH or ICSH, which stimulates the Leydig cells, and the Leydig cells stimulates the production of testosterone, the male sex hormone, and this will promote the functioning of reproductive tract and the other organs. And let us see, the anterior pituitary, when secretes FSH, follicle stimulating hormone, it stimulates the Sertoli cells. Sertoli cells, it stimulates spermatogenesis, and we know that these Sertoli cells, into the Sertoli cells, the sperms are embedded their head, and they transforms into adult by a process called spermiogenesis. Inhibin will be released from the Sertoli cells, which give a feedback mechanism. That means it inhibits the secretion of hypothalamus, that is GnRH and FSH and LH. Then the activity of spermatogenesis, spermiogenesis, etc. are controlled. Here also, released factors inhibit the LH and GnRH production by inhibiting or giving the feedback mechanism to the hypothalamus and anterior pituitary by inhibiting their secretion. So that's about the hormonal control of spermatogenesis. In next class, we'll discuss the process of oogenesis. Very soon, we'll see in the next class. Thank you.